So we're going to dive into this incredible story of science and strategy. You've got this devastating plague just tearing through the world's largest pig population. And all the old methods, they were failing, and failing spectacularly. But then, scientists found a new way to fight back. Not with a sledgehammer, but with a scalpel. And that really gets to the core question, right? When a disease is on the rampage, how do you actually stop it without causing massive destruction? The traditional answer is often, well, pretty brutal and super costly. But what if there's a smarter way? This was the high-stakes problem researchers were facing in China. To really get the solution, you first have to understand the villain of the story. And in this case, the villain is a virus called African swine fever. Its arrival in China back in 2018, it triggered a full-blown crisis. African swine fever, or ASFV for short, was just this terrifying mix of problems. It's not just contagious, it is incredibly deadly to pigs. And the real kicker? There's no vaccine. So when it hit China, which is home to about half the world's pigs, it wasn't just a problem, it was a potential catastrophe for global food security. With no vaccine on the table, the only tool in the playbook was this grim strategy called stamping out. And the policy? It was simple and it was brutal. If even one single pig on a farm tested positive, the entire herd had to be called. It was a blunt instrument, designed to stop the spread no matter the cost. But this approach, it turned out to be a total disaster in China. Why? Well, you have to picture the landscape. You've got this huge number of farms all packed tightly together. Most of them are small-scale operations, and they're all connected by these unbelievably complex transport routes. Trying to stamp out the virus here was like trying to put out a forest fire with a water bucket. It was just economically devastating and, honestly, totally ineffective. So, the complete failure of the old way. It really forced everyone to think differently. Researchers knew they needed a smarter, way more targeted strategy. And the breakthrough? It came from a crucial observation about how this particular virus was actually behaving on the ground.
You know, researchers on the ground noticed something a little odd. Unlike other agricultural plagues that can just rip through a herd in a matter of days, they saw that ASFV was actually moving much more slowly. It wasn't this big explosive chain reaction. And that seemingly small detail, that was a brilliant insight. It revealed a weakness they could actually exploit.
This one insight led to a complete radical shift in strategy. Think about it this way. The old stamping out method was like dropping a bomb to hit one single target. The collateral damage was just immense. This new idea, which they started calling precision removal, was much more like using a sniper. Surgical, targeted, and designed to neutralize the threat without destroying everything else around it.
So this sniper approach sounds great in theory, but how does it actually work in practice? Well, it works through a systematic three-step process that combines intense surveillance with some really rapid, high-tech testing. Let's break it down.
Okay, the whole method really rests on three key pillars. First, you've got comprehensive sampling to find every last trace of the virus. Second, you have incredibly fast and accurate DNA testing to get results in hours, not days. And third, you have the surgical removal of only the infected animals, all based on strict, data-driven rules. And when they say whole herd sampling, they absolutely mean it. This isn't just a few random checks here and there. No, we're talking about taking swabs from every single pig, but also testing the environment itself, the surfaces, the feeders, the water troughs, even the staff. They wanted to create a complete 360-degree picture of where the virus was, and just as importantly, where it wasn't.
Now, the engine that powers this whole strategy is a technology called qPCR. You can kind of think of it like a DNA photocopier that's so incredibly sensitive, it can find even the tiniest little trace of the virus's genetic material. And the best part? It is fast. Instead of waiting days for results, they could get answers in just 3 hours. That speed is absolutely critical when you're fighting a disease in real time.
And finally, step 3, precision removal. So, armed with that super fast qPCR data, they could act with surgical precision. Instead of culling the whole herd like before, they followed really specific, data-driven rules. For example, if a single pig in one pen tested positive, only that pig and its immediate pen mates would be removed, leaving hundreds or even thousands of other healthy animals completely untouched.
So that's the theory, right? A targeted, data-driven approach. But the real question is, did it actually work out in the real world? Well, the results from their trials on 4 different farms show exactly how this new strategy held up under pressure.
So each of these bars you're looking at represents a different farm that was having an active outbreak. The height of the bar shows the retention rate. That's the percentage of pigs they actually managed to save using this new method. And the results? They were just stunning.
Seriously, let that sink in for a moment. In their final and most refined trial, they achieved a retention rate of 99.72%. They went from a policy that guaranteed a 100% loss to saving virtually the entire herd. That is an absolutely game-changing number.
And this is how they tracked their progress. These are detailed electronic maps of the farms, where every single square represents a specific pen or an area. If a square was red, it meant the virus was detected there. A green square meant it was clear. The whole operation became this methodical, data-driven campaign to track down every single infection point and, through precision removal, turn every last red square on that map to green.
And what's really crucial here is that this was a learning process. I mean, look at this. On an early trial at Farm 2, they saved 65% of the pigs, which was already a massive improvement over the old way. But by the time they got to Farm 4, they had refined their sampling, they had perfected their removal protocols so much that they saved nearly 100%. They just got better, faster, and more effective with every single trial.
Looking at the full data table here, you can see the two key metrics right next to each other. The retention rate, as we saw, climbs dramatically. But now look at the column that says time to clear virus. It drops from weeks down to, and this is incredible, just 1 day on Farm 4. They identified the virus, precisely removed the single source, and had the entire farm declared clear in 24 hours. Just amazing.
So, what is the big takeaway here? This isn't just a cool story about saving pigs in China. This research provides a potential blueprint for a whole new era of smarter, more efficient disease control, period. Because the principles behind this success, they're universal. It's about shifting away from these incredibly costly reactive measures and moving toward proactive, precision-based strategies. This has huge implications for protecting our global food supply, for ensuring economic stability, and for managing animal health in a much more humane and effective way. When you boil it all down, the success of this method comes down to 4 key principles. You need early and rapid detection from on-site labs. You need precision evaluation to map the entire battlefield. You make data-driven removal decisions, not ones based on fear. And finally, you have to be committed to constantly learning and improving the process.
And that leaves us with one last fascinating question to think about. We've seen how this precision sniper approach completely revolutionized the fight against a plague in pigs. It really makes you wonder, where else could this kind of thinking be applied? Could this be the future for how we confront the spread of disease, not just on the farm, but maybe far, far beyond?